Very good morning to all. Very good morning.
Good morning, sir.
Good morning. Good morning to all. What happened? My video is not showing. Background. What happened? What happened? My camera is not showing. Huh? What's going on here? Okay. My just a second. I will stop share and again I will check why it is not coming. Camera cannot start video. Cannot start video, no? Why? Just a second. I leave and again I join. Why my video is not coming? What happened? Fail to start video camera. Please select another video camera in settings. What happened to this? Why? Okay. Anyhow, we will start. Okay, let it be. My video is not showing here. Okay, no problem. We will start. Why this happen? Okay, leave it. We are discussing the viruses now. Virus. Already we discussed the viruses, virus general characters, and what happened to this? Okay. Here you can see we are discussing the what we are discussing here. Okay. Virus, viral general characters of the viral that we are discussing. Now, we already studied who first discovered the viruses that we have studied. Who is that? Who given the name of the virus? The Bergering. First named it as a Contagium vivum fluidum. Contagium vivum fluidum. And then first crystallized virus is the tobacco mosaic virus. It is causing the tobacco mosaic disease. And Frederick, I already told you this all. First to discover the group of virus which causing the disease to the bacteria that they named as bacteriophages. And I already told you Nall and Rusk first identified the viruses under the electron microscope. Ivanovsky first identified the viruses. Ivanovsky first separated the viruses, first identified the disease. The first viral disease identified the foot and mouth disease, which caused to the animals, foot and mouth disease. Foots and mouth infected with the virus. The foot and mouth disease is the first viral disease identified in the worldwide. Okay. And I already told you the different characteristics, living and non-living characteristics of the viruses. It having the living and non-living, both the characteristics are there. But at outside, it acting as a non-living organism. Whenever it enter into the host, then only it will be acting as a living organism. It totally occupied their enzymatic action of the host. Enzymatic action of the host, and we convert their enzyme activity totally, it get modified, their enzyme activities inside. But at outside, it is as like a crystal. There is no metabolic reaction, no enzymes, no cell organelles, no cell wall, no cell structure, acellular. Acellular, there is no cellular structure. But it having the both the character, that's why we call it as interconnecting link between the living and non-living organism. That's why we call it as interconnecting link between the living and non-living organisms. Interconnecting link. It having the both the living characteristics and non-living characteristics. Living characteristics are there only in the host. If it is present in the host, if it is outside, it will be acting as a non-living organism. There is no metabolic reaction. It will be like a crystal. There is no need of the nutrition, respiration, excretory, no need of it. And the classification also we have studied various type of the classification based upon the host, plant virus, okay, phytophage, zoophage, animal viruses, and then cyanophage, it survive on cyanobacteria, gymophage, which survive on yeasts, like that based upon the host also we have studied, and pages. And for ICTV classification also we have studied. These all we have studied already. Again, why not? We have a lot of syllabus is there. Then the Baltimore classification based upon the genetic material, based upon the nucleic acid, classified into 8 groups that we already studied. Double stranded DNA, single stranded DNA. Plant virus are having the RNA, and animal virus having the DNA as a genetic material. And some of the plant virus also having the DNA, and some of the animal virus also having the RNA. That also we have discussed. And then Based upon the symmetry also we classified, that is icosahedral symmetry and helical symmetry and complex symmetry. Three type of the symmetries we have studied. And then we also studied based upon envelope. I already told you envelope is present only in the animal viruses, not in the plant viruses. The symmetries which we have studied, helical, helical symmetry and icosahedral symmetry and complex symmetry, both the type of the symmetries which are in. And the various viruses, its shapes also we have seen, we have studied. And sizes of the, the smallest, these all are smallest than the smallest bacteria, smaller than the very smallest bacteria. The chemical composition, it having the only nucleoprotein. There is no other chemicals, no other cell organelles present. Only it having the nucleic acid inside, it surrounds with the protein. That's it. No other extra organisms are present. But in the animal viruses, envelope is present, extra envelope is present. That is made up with lipopolysaccharides. Okay. The core protein which are covering, that we calling it as capsid. You see, nucleic acids, either DNA or RNA, but both are not present. DNA viruses or RNA viruses. Double stranded DNA, single stranded DNA. Double stranded RNA and single stranded RNA. Both the types are present. Okay. And the other than that, only the nucleic acid RNA is present, that we call it as virides. The whole organism of the virus we call that virion. Whole organism of the virus, that means nucleoprotein, we calling it as virion, that attack the living beings and it invade into the living beings, it causes the diseases. And then the next is what it is, prions, means only protein layer is present, only the protein layer is present, no nucleic acid inside, only the protein layer is present, that we call as prion. With the prion only they identified foot and mouth disease in the animals. Okay, with the nuclei, that means the viri, that mosaic disease, potato mosaic disease, potato spindle tuber virus, that having the only nucleic acid material, no protein code. That also there. Virion, viride, and prion. Virion means total nucleoprotein, nucleic acid plus protein. Viride means only nucleic acid, either DNA or RNA. Okay. And then prion means only the protein code, there is no nucleic acid material inside. Okay. And we also studied isolation and purification of the viruses. For the isolation, we have to identify the virus first, clinical specimen, what type it is, what type of the disease it is causing, where the symptoms, either leaf or stem or bark or root or where, how the multiplication is taking place, that we have to know the effect on host, and also preparation of the vaccine. We need to isolate and purify the viruses. The coronavirus, COVID-19, very dangerous, tragic years, 2 years, because of the virus, coronavirus, that caused so many deaths, so many people are died, so many people are affected, so many families and economy, world economy also get affected with this coronavirus. Like that, at that time, what they have to do, they have to identify and purify, isolate the virus. Based upon that isolated genetic material, we can prepare the vaccine for the fatal virus diseases. There's so many viral diseases are there now. What are those? Even AIDS, polio, SARS, influenza, so many diseases. Even in the plants also, so many diseases which causing by the viruses. Virus means poison. Virus is causing so many diseases to the plants, animals, and human beings. There is no usage of these viruses. These are majority, these are harmful. For that, what we have to do by isolation? First we have to select the infected plant material and extraction and infectivity test, whether it is infecting or not, that we have to test, and purify and virus shield. Again, we have to multiply these viruses and storage the purified viruses and use the purified viruses for further studies. Okay, virus propagation in the suitable host plant. You know, propagation host, especially Arachis, Beta, Cucumes, Cucumber, Pisum, Ziamage, Vigna. The selection of infected plant for first, that is the first step. And then what are the factors it is causing for the disease, and extraction of virus using the suitable buffers. Various buffers are there for the extraction of various pH levels. You have to use the various pH levels and various cations like calcium, magnesium, and then various proteases that we have to. What are those methods? We have to heating by centrifugation, by crystallization and salt precipitation, and precipitation at isoelectric point, and co-conservation, gel filtration, and density-based electrophoresis. By heating, we can extract the stable virus TMV that the crystallized, first crystallized by Stanley. You see the crystallization and then centrifugation. In the different speeds, we can separate the protein and nucleic acid so that we can separate the nucleic acid material and proteinaceous material, and we can separate the host material from the virus material by different centrifugation, gradient centrifugation, and precipitation at the isoelectric point, and also co-adsorption by separation of macromolecules in the liquids, and also by gel filtration, Sabadax or agar gel filtration, and also by density gradient method. These methods we can isolate. You can isolate the viruses, and then we have to do the test, in vitro test, whether it is infecting the host or not, how much it is infecting, and then so many methods already told you about the purification, gel electrophoresis, and co-conservation methods. These are the whole steps. These are the whole steps which are there in the purification and isolation and purification of the viruses from the plant, infected plant, to the separation of the viruses. These all are techniques. With this, we can purify viral protein. And the next, what I told you, the replication, transmission, and diseases caused to the plants, animals, and human beings. This we have to study. That all, what we have discussed is now that all are completed. Now what we have to study, replication in virus. The reproduction which is there, which is not there in the viruses. Why? There is no cell organelle, there is no reproductive structure, there is no specific organ structures are not there. Only the nucleoproteins are present, genetic material is present. Here, the multiplication of the genetic material is nothing but replication, multiplication, doubling of that genetic material is nothing but the reproduction we're calling here. As we already studied the bacterial reproduction, that also nothing but actually not the actual reproduction. There also there is no sexual organs are present in the bacteria. There also genetic material is exchanging from one organism to other organism. That's why we call it as reproduction, sexual reproduction. But here also there is no sexual organs, there is no cell structure, especially no cell structure, acellular. But we have studied the transduction, conjugation, transformation, transduction in the bacteria. The transduction mechanism same as the replication of the viruses. You know, bacteria phage which attack on the bacteria, T2 bacteria phage, and it inject the needles so that the viral DNA, viral genetic material enter into the bacteria, and the protein coat will be remain outside. That protein coat we call after injecting the viral genetic material, that we're calling it as what it is, ghost. What it is, the ghost. Okay, ghost. Eh? The genetic material is gone, and the remaining, that means as like a dead body. Whenever the genetic material is gone, the protein material is like a dead body. That's why we call it the devil, ghost. The genetic material that enter inside and that totally infect the host genetic material, and the host genetic material will be degraded, and only viral genetic material is multiplied. You know the why the virus are infecting the host? The basic reaction I told you, reverse transcription. Usually transcription means from DNA to RNA, from DNA to RNA. From DNA to RNA formation is the transcription in the protein synthesis, central dogma of protein. From RNA to protein is the translation. RNA to amino acid protein formation. Okay, this transcription is reversed in the viruses. That means from RNA to DNA. From RNA to DNA. RNA protein, RNA, which is modifying into the DNA so that it their own genetic material will be multiplied in the host. The host genetic material will be degraded, and viral DNA will be multiplied. And then whenever it multiplied, whenever incubation is complete, then all the virus will come out by splitting the host cell. If these all are coming by splitting the host cell, by breaking the host cell, that is lytic cycle. If it is associated with the bacterial genetic material, that means associated with the host genetic material, that we call as lysogenic cycle. These are the two type of the replications are there that we are studied. Okay. Okay. Yeah. What are the steps are there? Attachment is attached to the host, and penetration, entering of the DNA inside, and uncoating whatever it is the genetic material that uncoat, and it multiplies inside and assemble the protein coat with the DNA genetic material, and whenever it fully formed the new viruses that is ready to release, that are ready to release, that by breaking the host cell, by breaking the host cell. Okay, these are all the steps. You know, temperate pause, virulent pauses. If it is causing the death of the host by releasing the offspring viruses, that is called as virulent pauses. If it is along with the host DNA, that is we call as prophage or temperate pause. Okay, these are the two types you see in the replication. First is attachment. Attachment of the virus, that means bacteriophage, to the bacteria. Whatever it is the virus that first attached, and then whenever it is attached, then automatically it inject. By injecting, what happened? Whatever the virus genetic material that goes inside, viral genetic material that goes inside, and you can see here you can see clear the lytic and lysogenic cycle. You see here the first step, what it is attaching, bacteriophage is attaching to the bacteria host cell. Okay, and then what you see And then what happened? What happened? The viral genetic material which are enter inside. You see the viral genetic material which are entered inside, and here inside the viral genetic material is multiplying, replication is taking place. Large number of the more number of the viral genomes will be generated inside, and bacterial genetic material will be degraded. Only viral genetic material will be generated, and also the protein code also will be generated. Whenever the genetic material and protein codes are prepared, the genetic material will enter inside the protein code and it form the new bacteria pages. And then whenever it fully loaded with the new viruses, that means the bacteria pages, it splits. It splits the bacterial cell wall so that it releases the new bacteria pages, no viruses, no bacteria pages. That pages we calling it as virulent pages. It's causing the death of the host. The new offsprings, these are coming by death of the host. That's why we calling it as virulent pages. So many competitive exams they ask the question. The virulent pages are released in lytic cycle, lysogenic cycle. It's lytic cycle. After breaking up the host cell, the new viruses which release out, that we call it as virulent pages. Okay. Whereas in the lysogenic cycle, what happened here? Here you can see the whatever the viral genetic material which enter inside, that viral genetic material is intact, attached. Viral genetic material is attached into the bacterial genetic material, is multiplies along with the bacterial genetic material. It multiplies along with the bacterial genetic material. So you see here it multiplied along with the bacterial genetic material. Whenever the bacteria is taking the fission, binary fission, it forming two nucleic acid materials. In the two blue color you can see here, there is the viral genetic material. The brown color is the bacterial genetic material. Viral genetic material is mixed with the bacterial genetic material, intact, attached with the bacterial genetic material, and it will be continued for the next generation. You see here the viral genetic material, and again it participates in the another lysogenic cycle like that. It is forming a transformation of the genetic material from virus to the bacteria. But here it is not killing the host. It is the viral genetic material is continuing along with the bacterial genetic material. Viral genetic material is replicating along with the bacterial genetic material, is not causing any death, any damage to the host bacteria. These are present in the lambda page bacteria. And the lytic cycles are present in T2 page bacteria, and lysogenic cycle is present in lambda page bacteria. These are the lytic and lysogenic cycle. Lytic and lysogenic cycles. You can see here, this is the lytic cycle, this is the lysogenic cycle. Lytic cycle propages new pages, we call it as virulent pages. Lysogenic cycle pages, new pages, what we are calling it as temperate pages, which are not causing any death damage to the host. Okay, this is very important. Replication in the viruses. What are the steps are there? Attachment, penetration, incubation, that means replication, incubation, and release. These are the steps are there. These 4 steps. Okay, very important, the replication, and then transmission. Next, we will discuss the transmission of viral diseases. Next is transmission of viral disease. Here you can see the how the virus are entering, penetrating inside, viral genetic material is entering inside and is multiplying in the bacteria or whatever it is the host cell are getting damaged, and the viral genomes is multiplying and releasing one virus enter inside, and it releasing multi number of the viruses, new viruses outside. That multiplication is calling it as replication. Here, genetic material is multiplying. You see in the HIV life cycle how it is entering into the uh, HIV virus inside, how it is multiplying, how it is degenerating in the proteins, how the viral genome material is multiplying and it releasing into the body. Okay. And then the next major question in the virus is transmission of viruses in the plant. The transmission of viruses in the plant, okay, transmission of viruses in the plants are basically vary in the various level. These are, the virus are non-living, as like a non-living structure at outside in the environment. If it is outside, it can infect all the plant, animal, and human beings. Even not only human, even bacteria, cyanophyceae, yeast, or these all, even fungi also will be affected with these viruses. I already told you, viruses are the obligate intracellular parasites. This can survive only in the host. If host is not present, they can't survive. We, I already told you parasite type, obligate parasite and facultative parasite. If it is survive only in presence of the host, in the host, that we call it as obligate parasite. Even it can also survive at outside without the host, that we call it as facultative parasite. But all the viruses are obligate parasite. All the viruses are obligate intracellular parasites. It enter inside the cell and it multiply the genetic material and replicate the DNA or RNA. DNA and RNA replication will take place. That means reverse transcription will take place. Okay. So there is a lot of chance for the viral infections, either horizontal or vertical. That means either by vector or non-vector. Vector-borne viral diseases or non-vector-borne viral diseases. Like that, if it is depending upon the vector, that are vector-borne diseases, especially transmission of plant viruses by mechanical transmission. The mechanical transmission means if by touching the infected plant material to the healthy plant material, by touching the infected plant material with the healthy plant material, that we call it as that we call it as okay, infected plant material to the healthy plant material, that we call it as mechanical transmission. Mechanical transmission. Either the plant extract, either the plant extract or the plant material or infected plant, if it is infected plant, if it is touch attached with the if it is attacked with the healthy plant, the healthy plant also will be get infected. That's why, you know the first early stage only we have to remove, and early at as much as early stage we have to remove the infected plant from the field. Otherwise, it spreads within the short span of the time. It causes a large damage to the all the field. Whenever we identify one single leaf or single leaflet, then we have to immediately pluck it and fire it. That is the best quarantine method of the viral diseases. Okay, and by vegetative or graft transmission, by vegetative propagation, if we do the just a second, if we Okay. If we do the transmission grafting or whatever it is the mechanism, if we do the grafting mechanism, uh, a vegetative propagation mechanism, at that time, if we graft from the infected plant, viral infected plant, the by rhizome, you see ginger, like the turmeric, or by bulbs, uh, onion, whatever it is, garlic, and by corn, uh, tubers, potato, that all. By doing the vegetative propagation, we have to take care. We have to sterilize with the viral virescent. That way, that virusides will be prevent from the diseases. Okay, and you know the pollen transmission. By the pollen transmission, also pollens also, the viral may attack upon the pollens. That pollen will be caused whenever it pollen it may enter into the another flower stigma. That flower, that plant, other plant also will be infected. And seed transmission. Seed, if the seed is infected with the viruses, that seed again, whenever it germinates, the new plant also will be infected with the same viruses. You know the nematodes which are there in the soil, nematohelminthes, platyhelminthes, and nematohelminthes, especially Japanima and Languidorus nematodes, which are there in the soil earth. By moving it in the earth, it will, whenever it attaches to the roots of the plant, that roots will be infected with the viruses. And a fungal infection, fungal transmission also is there, especially Olpidium brassica on the brassica. It causes the disease, Olpidium, which are associated with the viruses, so that it will be transmit the viral disease to the healthy plant. And insect vector transmission, especially majority of the viral infections is caused by insect vector or aphids, moths, thrips. Every day, suck the juice from the infected plant, and again it goes to the healthy plant. Again, it goes to the healthy plant, so that what happens? The healthy plant will be attack the viruses. Why the whatever the juice extraction, the juice extraction extracted from the infected plant by the aphids, and again it goes to the healthy plant. Then what happens? The healthy plant also will be infected by the juice having the viruses. You know, Contangium vivum fluidum, Bajering said, we can't filtrate with the bacterial filter. These are very smaller than the smallest bacteria. So that fluid, it causes the disease to the healthy plant. And you know, another one is the dodder plant. You know the Casquita, Bangaru Tigandam, yellow color upon the plants. Upon the plants, you might have seen the yellow color climbers, yellow color finner, creepers. That yellow color totally occupy the plant. The dodder plant, parasitic plant, it having the viruses, so that all the host plant will be infected with the virus. Along with the parasitic plant, it also being infected with the viruses, so that all the host plant will be died if the dodder plant attack on the healthy plant. Okay, and then here you can see the various vector, nematode vector, insect vector, by insect falling upon the infected plant to healthy plant, or mites or trips. You see how it is entering inside the vector feeding, so that by feeding upon other healthy plant, it is going to the next. You see the natural transmission, the intracellular and intercellular or extracellular environment, it will be transmit from one to other, and from one cell to all the cells. The common vectors are, what are those? Bacteria, fungi, nematode, arthropods, and arachnids. Okay, the our human being infections out by grafting, by touching the infected plant and healthy plant, and a mechanical touch, so that, and the transmission between the plant cells. Whenever it attack one plant cell, automatically if it widespread, the systemic diseases, local diseases and systemic diseases. The local disease. What is the local disease? What is the systemic disease? Local disease means if it is infect only one organ, one part of the plant or animal, whatever it is, that is local. Usually, majority of the plant viruses are systemic. That means it infects all the plant. It infects all the all the living organism will be infected. All the living organism will be infected. All the part will be infected. That's why we call it as systemic diseases. Okay, and you see the types of cause of diseases. Airborne diseases by the air. It may be spread from one to other, or foodborne diseases. Okay, bacteria, toxins, viruses, prions, and parasites. And by the infections, contagious diseases. That means from one to other, one person to other, especially influenza. The influenza, polio, so many are there, na? And communicable diseases, cholera, typhoid, polio, what are those? Hepatitis, rhabdo, rabies. The non-communicable diseases which are not widespread from one to other, non-transmittable, non-communicable from one to other. You can see the plant virus diseases. There's so many diseases which are necrosis, mosaic, vein cleaning diseases, and uh, chlorosis disease. The total chlorophyll will be damaged, infected, and total uh, uh, dead spots upon the plant, and also the stunting of the plant growth will be reduced. The diseases are again non-persistent, semi-persistent, and persistent diseases. If it is continue till the death of the host, sometime it may be semi-persistent. I already told you nematodes, Japanima languidoras, and plasmodiomasis, and seed and pollen also transmitting the disease. Direct plant to human transmission, and you see how the transmission is by the stomata or by the hydatodes or from the lenticels. These are the ways, passageways to enter the viruses inside. It causes the infections. Okay, and you can see the viral disease transmission. You see, I already told you virides. Virides means only the circular RNA, genetic material without the protein code. It caused potato spindle tuber virus. It caused potato famine in the Ireland. Okay, it infects all the potato plant crop. And you also can see the prion. Prion, it having the either only the protein code, no DNA or RNA, no nucleic acid material, only protein code that cause the mad cow disease, foot and mouth disease or mad cow disease. You see the foot and mouth are get infected. They can't stand while eating the grass. Also, it infects the mouth, and also touch the whatever the viral infected area. It cause infected the legs, foot, and the mouth. Foot and mouth disease, the mad cow disease, which is caused by prions. Okay, and then here you can see the plant viruses: tobacco mosaic virus, tomato mosaic virus, potato virus, papaya mosaic virus, cucuma virus, and you see tobacco necrosis virus and cauliflower mosaic. Various viruses are there. Based upon the viral infection, Baltimore, as per Baltimore classification, you see Sarcoviridae and Geminoviridae and Cornoviridae, Luteroviridae and Sicoviridae. Formaviridae types, they classified based upon their infection status, their abdat, their infection stages. Okay, here you can see uh, where it is viral diseases in the plant. Viral diseases in the plant. What are those viral diseases which are causing? You see the mosaic disease, mosaic, multicolor. This mosaic disease, sometimes we are using for the benefit purpose also. In the tulip, tulip flowers, if it is infected with the viruses, it will be multicolor flowers. My voice is clear? Multicolor, so that the flowers will be attractive. And the leaf curl disease, leaf will be curled back, bend back, and vein bending, total vein, mid veins and side veins, total veins will be bent back, and yellowing of the veins in the bending, and leaf roll, leaf folded as round, and bunchy top disease, bunchy top of banana. Because of bunchy top of banana, banana crop damage in the Sri Lanka, bunchy top of banana. And vein cleaning, you see the whole vein will be yellowish. All vein will be in the yellowish in structure, yellowish in color. You see the transmission we already studied now by seed. You see so many viruses, BCMB, and we can see the Casquita. You might have seen the Casquita, Bangardiga, Dodder. They is called it's widespread on the big trees. It's having the virus also along with that, so it caused the death of the host plant. Transmission through the vegetative propagation by doing the grafting, the bulbs or corns or rhizomes. If these all are infected, the next plant also will be infected. The soil, especially in the some of the soil-borne disease, viruses also is there, potato mosaic and oat mosaic and wheat mosaic diseases. This viruses will be in the soil. Whenever the new plant germinating, at that time, this will be infect the new plant also. And then majority by insect transmission, insects by sucking aphids, lithopods, beetles, scaly insects, thrips, mites, white flies, by sucking the whatever it is the plant material, whatever the virus also enter inside, and again whenever it goes to the healthy plant, that healthy plant will be attacked with this plant, with this virus. And transmission by fungi also, I told you, Olpidium brassicae, especially in the brassica, vein mosaic virus. You see Olpidium, Spangospora, and Syncytium. These transmit viruses, transmit by nematodes, especially nematode, Japanima and Langidoras, that nearer to the roots of the plant, that cause the transmission of viruses, nipovirus, nitoviruses. Okay, nematode transmitted polyhedral virus, nematode transmitted tubular viruses. The both are there, that both infect the plant. Yes, we already studied attachment and penetration of the uncoating virus, replication and genome replication, multiplication inside, assemblage of the genome with the protein, and releasing of the virus. Replication, the plant viruses that we have studied. Management, we need to manage the aseptic condition. We have to pluck and fire the infected plant material very early, or otherwise virucide, dimethoate, and mercaptan, jet 78, and methyl parathion. By spraying these material, these chemicals, we can stop the spreading of the viruses, but we can't control fully. We have to collect the healthy plant seed material. We have to collect the healthy plant rhizome, bulbs, whatever it is, and we have to sterilize the agriculture tools before doing the flow work or sowing work or separation work or cleaning work in the agriculture. And then uh, sterilization of the viral genetic material, that also. And you see the genetic host resistance varieties. You have to prepare the host resistant genetic host resistance. You know the whole root tip, shoot tip having the highly resistant to the viral diseases. So if we do the transplant tissue culture, multiplication of the plant material by root tip or shoot tip, we can generate the viral resistant, viral disease resistant plants and control the vectors. Main thing is this one. We have to control the vector, mite, thrips, white flies, that all we have to control by spraying the insecticides, so that if the insects are not there, the viral disease will not be transmitted from one to other. And so many cultural practices, that means geographical isolation of the viral diseases. We have to maintain the distance between the plants. We have to make thorough the cleaning of the environmental conditions, and we have to make thorough sterilize the instruments before using in the field. Chemicals application, biological control, various chemicals, virucides and insecticides, and so many regulatory mechanisms are there. You see, these all are the some common viral diseases of the plant. These all are the viral diseases. You see, tobacco mosaic disease is caused by tobacco mosaic virus, TMV. It having the spirally coiled crop seed. It transmits through aphids or grasshoppers. Disease cycle, you can see the disease cycle infection. It transmits. You see how the mosaic virus causes tobacco mosaic. You see how it is. Animal and human viruses also is there now. So many animals also causing foot and mouth, mad cow diseases. You see transmission of animal and human viral diseases, how it widespreading by the animals, by the birds, insects, mice, thrips. These are spreading, transmitting the diseases from environment to animal, human beings, or men. You can see these all are the carriers for the viral diseases. You see, so many carriers are there, even monkeys, all animals, cat, snakes. These all are carry the viral genetic material. Birds carry the rats carry the genetic material, and if it is that viral attack material may be having touch with our human being, we also get infected. You know the rabies, that also viral disease is caused by dog bite, hydrophobia disease. You see the corona, how it affected the coronavirus, the globally, how it is globally spread the coronavirus. Uh, These all are we have studied, and then you can see here one more human diseases, no? Human diseases which caused by the viruses. So many human diseases. You can see the human diseases caused by the viruses. You know, these are, I told you, obligate parasite, different or use HIV, hepatitis, Ebola, influenza. Hmm. You see, HIV/AIDS is causing by the viruses. Human immunodeficiency virus. It's from chimpanzee origin. You see, the first report in New York, USA, 1981. You see the syndrome, AIDS, it's causing by HIV virus. And mode of transmission, you know, you already know, sexual contact, contaminated needles or syringes, and blood to blood contact during the blood transfusions, and mother to child, close contact between the infected and non-infected people through cuts or open wounds. Through cuts or open wounds, it cause or injection or physical touching or sexual contact, needle syringe using, and mother to child, it will be transmitted. Okay, and then we also know, and then we can see uh, just a second. Where is the gun? Okay, but is not transmitted by air or water, saliva, sweat. You know, we are all very aware about the air. Sharing toilets, pets is not transmitted. You see the HIV symptoms: fever, night sweat, mouth ulcer, fatigue, chill, muscle ache. You see, these all are the symptoms. The treatment, we all know, highly active antiretroviral therapy. Oh, time going to upper, but I will come to it. Treatment by secondary infection, and you know the polio, also viral diseases by poliomyelitis, polio viruses, and injection by food or water contaminated diseases. You see, virus in phase remain viable 3 to 4 months. You can see incubation period is 7 to 14, 1 to 2 weeks. How the polio infected people are. You see the bending of the legs and foot, hands because of polio. Partial paralysis will be happen. Polio vaccine. You see, Salk and Sabine. Salk and Sabine first discovered the polio vaccine, oral polio vaccine and injection polio vaccine. Now we are using the oral polio vaccine, polio vaccination. And you can see the chicken pox. First vaccine discovered is chicken pox vaccine, smallpox vaccine by Edward Jenner. You see how what are the source of, you see, skin, buccal mucus, and vascular fluids. These all are infected by red, chill, you see, blisters, pupils, ulcers, and treatment by Takahashi, Okastain, and also is there Pamiclo tablets. And you know the hepatitis, uh, it is inflammation of liver. You see, the causative agents, hepatitis virus, A, B, C, D, E are there. Type B and C lead to chronic disease in hundreds of millions of the people. You see, these people got the Nobel Prize for the discovery of hepatitis C virus. Who's that? Alter, Houghton, and Charles Rice. Hepatitis, it infects the liver. You see mode of transmission, hepatitis A and B by the ingestion of contaminated food or water with fecal matter. Hepatitis B, C, D through various body fluids. Through the body fluids only it will be infected. But A and E by fecal material or contaminated material. Okay, you can see the symptoms: fatigue, yellowing of the skin and eye, jaundice, stomach ache, and liver cirrhosis, and vomiting. Dark urine is very dark. These all are the symptoms. The treatment: hepatitis B vaccine is there. Hepatitis A, B, C vaccines are there. These all are vaccines we are using. And then naturally also we use the Pilanthus neruri, Pilanthus emblica for the for the yellow fever, jaundice. You see the rabies, dog bite disease is caused by dog bite. Is causing by the dog bite. You see here the dog bite by this dog bite by, to the teeth of the dog. It is entering inside, and you also can see rabies reservoir. You see the what are those? Not only the dog, fox, monkeys, and also oh, there it is. And here also you can see mode of transmission by brain inflation. Incubation, it will be incubated in the dogs, cattle, animals in up to 20 to 90 days. It transmitted through the infected wounds. You can see how it is. Oh, what happened? And then you can see the animal symptoms. Those who are infected with the rabies, their teeth and their mouth, how it is. And symptoms of rabies in human: fever, brain inflation, season confusion and coma. And you can see treatment: anti-rabies vaccine. You have to take it the stomach, no? And Ebola virus, you know the Ebola virus that caused famine. Ebola virus, Africa country to infected much of damage. You see by the bats, the Ebola virus is transmitted from bats to the human beings. And then you can see the various Ebola affected famine time at the and the you see the Jabo vaccine and remdesivir. Even Corona time also we use the remdesivir. You see rabies day, AIDS day, polio day. These all days we are doing. The next very recent coronavirus, this corona 1720 times plague, 1820 times cholera, 1920 Spanish flu, and 2020 coronavirus. These are all the famines caused by these viruses. Okay, how these all are infected, how the famine happen, the coronavirus. Okay, like that other viruses also. You see the coronavirus, COVID-19, we already, very recently, we all known. SARS, other SARS viruses. Corona also one of the SARS virus. Severe acute respiratory syndrome. The China 2003, it caused so many deaths. And MERS, Middle East respiratory syndromes, MERS COVID, this also caused so many deaths. COVID-19 pandemic, it started in the China, Wuhan, the 2019. You see how with the symptoms we all know very well: fever, cough, muscle pain, headache. How the transmission of the corona, we all know very well about it. You see corona preventions, this all we all know very well. Okay, these all are the human diseases which caused by the viruses. Plant diseases caused by the virus, human disease caused by the virus, and animal diseases, and transmission of the viral diseases, and also regulation control of the viral diseases. Today we have studied replication, transmission, plant diseases caused by virus, animal diseases caused by virus, and their control measures. These all we have studied. These all are essay questions. Replication, the virus essay question. Transmission of viral disease in the plants, essay question. Controlling measure of the viral disease is the essay question. Plant disease caused by the viruses, essay question. Plant disease caused by animal disease caused by the viruses, essay question. These six essay question. With this, we have concluded all the syllabus we have completed. You go through this all the videos so that you can make it repeat, you can learn, you can understand by repeating it again in the next. Okay, so that you can learn, you can repeat, you will go through these all the videos, all the syllabus, 100% syllabus covered in our 20 classes. You go through this all the 20 videos, you will go thoroughly, and you will get the full marks. You can write accurately, you will get the full marks. Wish you all the best for your exam. Okay, you all go through these videos again. Whenever you have the doubt, you go to the YouTube, BRAU, and you open it and you watch again so that you will get clarify your doubts. If you have again any doubt, you can ask, you can message, you can ask at any time. No problem at all. Okay. Wish you all the best for your exams. Okay. Okay. Is it okay? We have completed all the syllabus. Virus, bacteria, fungi, general character, classification, reproduction, nutrition, classification, importance, uh, fungal disease caused to the plants and human beings, and also what you have studied, the fungi, economic importance of fungi, and the role of food, agriculture, industry, medicine, and harmful effects of the fungi, and mycorrhiza, mushrooms. This is all we have studied. Okay, this is all your syllabus that we have completed. Okay, good, nice. Wish you all the best. Take care. Okay, bye.